evolution to it. Okay. And we have biotech, both principles and applications combined. Principles plus applications. And then we have ecology and biodiversity. So let's keep it together. Majorly, am I missing anything? Wait. Yes, so we have reproduction, genetics and
Human health
evolution,
and microbes.
biology and human welfare, right? Which
Yeah.
Yes.
it had two chapters, no? It had the human health and disease and microbes in human health welfare. Yeah, right. So
Yes.
it comes, sorry. Yeah, sorry. Why is this? Yeah.
So reproductive health comes
We
under human health and
have,
disease category, right?
let's put it in reproduction. Reproduction plus reproductive health, right? And then human health and diseases. We will not focus in human health and diseases. We will major thing is about, malaria and other things that we did, right? The mechanistics. But in reproductive health, we talked about specifically about art, assisted reproductive techniques, and how certain diseases can affect just the reproductive system and stuff like that. And then microbes, we talked about microbes in human welfare. Okay. So what we'll do is we will start. Okay, yeah, let's start with Parker. Parker, what's your take
Yes.
on this? What do you think is what do you think is the best for you? What you
Definitely
understood the best? Yeah, tell me.
reproduction plus reproductive health.
So for Parker, it was. Let's also do one thing. Let's number it so that it's easier for us. First, second, right? Broadly 5 things it is divided in. Yes, Parker. For you, it is?
One.
Okay. So you have one, which is, let's talk about strength. So in your strength, you think it's 1 and?
Probably 2.
And 2. So you are sure with reproduction, reproductive health, genetics, and evolution. Okay. And what do you think is your weaknesses?
No. Strength is 1 and 3 probably.
1 and 3, which is human health and diseases and microbes and human welfare.
Yes.
Okay. And so other 2, 4, and 5, where will you put it?
5 is in weakness.
Okay, so let's talk about weakness. 5, you'll put in weakness, okay?
And I think strengths, biotech can also go in strengths.
You're sure, a little sure about biotech as well, right?
Yes.
Okay, so I'm putting here as 4, 1, 3, and 4. What about
Yeah, then
genetics and evolution? Where do you yourself?
5 and weakness.
Okay, so it's 2 and 5. Okay, what about you, Iram?
Um, Strength B1.
And I'm discussing it in the class because now we need to plan in a more, what do you say, hybrid mode or more amalgam mode, because we'll be doing question answers. So I'll
Yeah.
keep these things in mind, and I'll also know that if something is strength for both of you, then I know that all I need to do is sharpen your speed there. But if something is weakness for both of you, or others also, then we will revisit some concepts again. Yes, Iram. So what do you think? Strengths.
1.
Okay, we also have Aisha joining in. Hi, Aisha. Sorry, I missed you there. How are you doing? Can you speak, Aisha? Okay. Yes, Iram. Go ahead. Strength is 1, which
1.
is good. Yeah.
4.
4,
5.
5,
3.
3 as well.
Yeah.
So 1, 3, 4, 5. 1, 3, 4, 5.
Yes.
So you are also categorizing genetics and evolution,
Yes. Sir.
the weakness, right? You and Parker has genetics and evolution as common. Like, you think when I say weakness, it's not like you have not understood anything. We are just being a little more stringent here in calling things as strengths and weakness, because I want to be 100% sure that you are 100% confident about this. So whatever you are not 100% confident in, you can just blatantly call it your weakness, and we will work on it together. So as of now, 2 seems to be a very important thing. Yes. Is that it, Iro? This is the final
Yeah.
thing for you as well? Okay.
Yeah.
Okay, cool. How are you, Aisha?
So
Can you?
also one
Yeah.
more thing. So in reproduction unit, the first chapter, that is sexual reproduction in plant, it's a bit tough for me.
Okay.
The concepts.
The concept. So should I then, let's, for the sake of like for planning better, let's keep 1 in your weakness. No need to worry about how it will look or something like that. Okay, it's just for our purpose. Will it
Okay.
work?
Yeah.
Yeah,
Okay.
you okay with it? Okay. Aisha, how are you doing? Okay, Aisha is not able to speak up, so she's writing in the chat. Aisha, this is high time we should start talking because we are now going into a mode of less jargons and more work. Work in the sense you're going to spend more time doing questions. I would like to hear from all of you what you think, what you understand. Okay, anyway, so Aisha is writing in the chat that for her, the strengths are 1, 3, and 5. Okay, 1, 3, and 5, which is reproduction, reproductive health, microbes, and ecology and evolution. Okay, and then you are keeping 2 and 4 is kind of a little bit. You're not 100% confident. Yeah, so I will call it weakness, though it's a very strong term, and I believe that even in genetics and in biotech you understand a lot, but let's keep it that way. Okay, cool. So now, since we understand few things. So 2 is something, genetics and evolution is something that all 3 of you thinks that we should revisit some concepts. And after 2, is basically 1, 2, 4, and 5. So 1, 2, 4, and 5. So, okay, cool. So let's, as we discussed in the last class as also, let's target first in our concept building reproduction, like 1 and 2. We will keep reproductive health a little aside for now because those are mostly things you have to remember. Once you understood it, so once you understood what is IVF and um, what kind of diseases affect just the reproductive system and stuffs like that, then you just have to remember it. But by reproductive, I mean the mechanisms in plants and the mechanisms in animals, which is human centric, and then genetics and evolution. Okay, is that fine? Cool. So
Yes.
one way to do is let's do like this. So instead of before going to some questions, bad questions, let's also I'm not able to move things here. So I'll stop sharing this for some time and I will start sharing the note textbook. So can you all come to open your textbooks? Let's go in a series and try to narrow down the topics that we have to revisit. Can you all see the screen that I'm sharing? Yes, no? Everyone, can you see the?
Yes.
Okay, perfect. So because all of you start with flowering, yeah. Let's start with sexual reproduction in flowering plants. And you have to tell me which topics you find a little difficult. I'm just making a personal note of all of this. No worries, Parker. That's why I'm just showing the textbook here. But from now onwards, do two things. Keep your textbook and a notebook with a pen handy, because I might give you some questions here on board, and then I'll ask you to solve it just to see how fast and how accurate you are solving. And those will be the actual questions asked in exams. And that will also help me to see if that topic requires more concept building or not. So keep these two things. For today, you just need a notebook and a pen. Okay, so here, I think about the structure of the flower. Is everything clear with respect to the structure? And then we divided the whole event of reproduction into 3 parts: pre-fertilization, fertilization, and post-fertilization. Correct? So anyone has issues with understanding the structure, stamen, stigma, microsporangia, macrosporangia, like conceptual issue or difficulty? If you have not revised it, and that's why you feel that, okay, if I'll revise, I will again remember it back, then it's not an issue. It's just the lack of revision. That's not a major problem. But if you missed something when we were teaching in class, this is the time you can ask again and we'll go over it. If there are gaps in concept. Yes, I think Parker unmuted herself. You were saying something?
No, sir.
Okay, so anyone has issues with pre-fertilization event, uh, uh, structure and
No.
events? Let's first talk about structure. Yes, Parker.
No.
No?
Yes, I know.
Okay. No one. So if no one will speak, then I will assume that you don't have an issue. Aisha, you have to write then actively in the chat if you can't speak. Iram, any issues with the structure? Male and
No,
female
sir.
both? No? Okay. So if randomly something comes from anywhere like the tapetum and all, I'm assuming you all have understood it. We just need to go back and revise, right? So do this straight away. So Iram, okay, I'll come maybe. Any issues with the pollen, structure of pollen, and how it carries two male nucleus? Yes, Iram.
No.
No. Okay, what is what is the concept of vegetative cell? What is the concept of generative cell? And when a pollen tube forms, how generative cells and vegetative cells move and then go and fertilize the embryo sac. So any issues with this female part, embryo sac, this structure? So this diagram often comes in both boards and in entrances also. It might come with something not labeled and you have to find the right label. So all these labels, so there are too many here from Hilum, and all these lines are going somewhere. So everything is clear. All these layers of a ovule is clear. Parker, for Parker, it's clear. Aisha and Iram. Yeah, it's a good way. You can give me thumbs up so that I understand that it's all clear. Okay, perfect. And then rest was this thing. When fertilization happens, during that, there has to be a this megaspore, from one single cell becomes a dyad, then becomes a tetrad, and finally the most um, mature structure is a seven-celled eight nucleus structure. So everything is clear with respect to this seven-celled eight nucleus structure, which cell is what and which cell does what. Parker, Aram, Aisha. Feel free to say if anything is not clear to you and we'll revisit if there are lacks in. But if you feel that I will revise and I'll get it. I attended, I understood. If I'll revise, it will be all back, then it's fine. Okay, perfect. I see all three thumbs up. Then the event actual, the pollination. We talked about pollination and the kinds of pollination, self-pollination, cross-pollination. Basically three types: auto, Gitono, and xeno. Everything's clear between auto, Gitono, and xenogamy. Yeah. So these are three types. And then there are two different types of flowers: chasmogamous and cleistogamous. So which type of flower prefers which type of pollination, that part is also clear, right? For example, if it's cleistogamous, it will mostly go for xenogamy and why, sorry, mostly go for autogamy and why. All this is clear. Cleistogamous is closed flower. Okay, so for Parker, it's clear or not clear? I see a thumbs down.
No, sir. If I have to, I kind of forgot.
Okay, so do you remember we talked about pollination? Three types of pollination. I
Yes,
think Iram, you understood, right? Iram?
Yes, I understand.
Okay, so to just bring Jolt Parker's memory back, why don't you quickly tell about, in brief sentences, what is autogamy, gitonogamy, and xenogamy? If you want, I can also share the class notes, or if you're fine with the textbook, you tell me. So what was autogamy?
Yes, sir. So
Yeah.
there are kind of three kinds of pollination: autogamy, geitonogamy, and xenogamy. In autogamy, the transfer of the pollen grains is from anther to stigma of the same flower, and
Same flower, yes.
in same flower. And for this, the flower requires synchrony in pollen release and also the stigma receptivity.
Right, correct.
And the anther and the stigma should lie close to each other for the self-pollination. Example of these plants are oxalis and Viola commelina. etc. And again, in autogamy, we have two types. We have chasmogamous and cleistogamous. Chasmogamous flowers are which are similar flower of other species with exposed anther and stigma, and in cleistogamous flowers, the stigma is closed; it's not open, and the anther and the stigma are present in within the flower.
Okay. Right. And so and clistogamous flower go for, was that part clear? Iram, to you?
Sorry, sir, your voice was cracking.
Oh, sorry. I was saying, so this concept of chasmogamous and clistogamous flowers, that's clear to you, right?
Yeah.
So which type of pollination they will prefer? So if a flower is chasmogamous, which means it is open, right? Exposed anthers and exposed stigma, both. So
Yes.
what kind of pollination is possible in chasmogamous flowers?
Um, Cross pollination.
Okay, cross pollination. So if it is open, of course, cross pollination can happen, but can't self
It can't
pollination
happen.
happen? It can't happen in chasmogamous self pollination.
It can happen. It can
It can
happen.
happen, right? Both can happen because it's
Yeah.
open, exposed. So
Yes.
the pollen grain from either the same flower or from any other flower of the same plant or any different flower of a different plant, anything can land on the stigma because it's open and exposed. Correct?
Yes.
Yes. So do you understand, Parker? Are you following it? Basically, we are revising this for you. Okay. Now, if it is cleistogamous, in which the flower does not open, which type of pollination is possible and which type is not possible? You know,
Sorry, sir, I didn't hear that. Could you repeat?
in cleistogamous flowers, where the flower does not open at all. Okay. So
Yes.
which type of pollination is possible and which type of pollination is not possible?
Yeah. So since the flower is closed, only self-pollination can occur.
Right. Only?
Cross-pollination can't occur.
Yes. Right.
Self-pollination.
Only self. So the limit is with cleistogamous because it is closed and does not open. Okay. So it's very obvious that only self-pollination can occur in cleistogamous flowers. Right. We also discussed that just because chasmogamous flowers are open and their anthers and stigma are exposed, it does not mean that all the time, like because there is a possibility of self-pollination. Self-pollination is something that plants try to avoid. Remember that discussion?
Yes.
And why would a plant try to avoid self-pollination?
Because that can lead to variation.
Yes. So because variations won't be there. Perfect. In that case, what do these plants do in terms of some strategy or modifications? Remember that? If while discussing you can remember all these things back, you are very well prepared. So that's why we are discussing. Parker, you can also
Yes.
pitch in now. Yeah, Iram, go ahead. What do these plants do to try and avoid self-pollination? Some strategies, but
So
ensure cross-pollination. Yes, Iram, go ahead.
so the pollen release and stigma receptivity, they're not synchronized, and
Mhm.
sometimes it happens that pollen is released before the stigma becomes receptive, or the stigma first become receptive, then the pollen release.
Yes,
um,
right. So what Iram is trying to tell here is that the plant will deploy the strategy of developmental delays or asynchrony in the development of the male and the female sexual part, the filament and the stigma, right? Correct. Anything else they can do? There were other strategies also. You remember any other strategy? Do you remember something like a superior ovary, inferior ovary concept? We discussed that also. So one thing which will for sure work, and plants of course do that, you're right, is uh, asynchrony, offset
So I
in the
remember that.
receptivity. Yeah.
So the production of unisexual flowers.
Yes, so that's also a good point. If you'll produce unisexual, of course, self-pollination can't happen, but still self-pollination can happen from the same plant. That's also self-pollination, right? Basically, gitonogamies transfer from different flowers, but of the same plant, correct?
Yeah.
So if you talk genetically at the level of genes, because now you understand one organism has one set of genes, one plant is one organism. So gitonogamy is functionally a cross pollination, but genetically it is self pollination only. Does it make sense to everyone?
Yes, sir.
Thumbs up. Yeah. So even if you start producing unisexual flowers, the chances that there still can be some self pollination at a genetic level. So, but you're right. If you produce unisexual flowers, the chances of self pollination go drastically down, and some other anther from some other pollen from some other plant can also land very much. But do you remember how some plants, when they have no choice but to produce bisexual flowers, then what they do is they change the position of ovary and stamens, right? So if the female part is placed much higher than the anthers, remember that I drew a diagram where the stigma was much higher placed than the anthers. So if anthers shed pollens, those pollens will fall down, but their chances that they will go up and fall on the stigma is less. So if you have a superior ovary, which is where the female part is higher than the male part, the chances that someone else's pollen will land on it is more. But if you have superior anthers, like anthers are so high that they bend down like crown on top of stigma, their self-pollination will happen a lot. Make sense? Does it make sense what I'm saying? I will,
Yes, sir.
or should I try to make it? Does it make sense to everyone?
Yes, it makes sense.
Okay,
Yes, sir.
perfect. So positioning. So first thing is, like Iram correctly pointed out, first thing is offset or asynchronization in the receptivity and pollen release and stigma receptivity. One first point, very crucial. Second point is try to make more monosexual flowers. And third is uh, positioning of the female part and the male part. These three techniques they deploy, the chasmogamous flowers deploy to avoid self-pollination as much as they can, and entertain or appreciate or encourage more of cross-pollination. Okay, perfect. So these were some questions, important questions from this pollination part. Then agents part, you just have to remember there are biotic and abiotic agents, and based on what kind of agent a plant uses, the structure of the pollen will be also different, right? Can you give some examples, Parker? Do you understand this part? That if a plant relies on water, so it uses a abiotic agent like water for its pollination, then what kind of pollen it should have? What should be the property of pollens? Yeah, go ahead, Parker. Just quickly, briefly, just tell us. What will be the characteristics of pollen grains if it is a water pollinated plant? Yep. Parker, are you there? Iram?
Yes, sir, I'm there.
Yeah.
Sir,
So what
can you?
kind? Yeah, my question was, what kind of, how can you say, just give some examples that depending on what pollinating agent a plant uses, there are changes in its pollen grain structure. If this question comes, what will you write?
Uh, there is um, a mucilaginous coating.
Mm-hmm. There is mucilaginous coating in which kind of plants?
Water pollinated plant.
Water pollinated. And if it is wind pollinated, then they will be very light in their structure and weight, right? They will have some structures that will help them be airborne so that they can go with the wind, right? If you have seen the seeds of
Yes.
dandelion, something like that. And if they are animal or insect pollinated, then they will have sticky pollens that just stick on the body of the animal as good as they can. Correct?
Yes, sir.
Yeah. Okay, perfect. So that's it. That's about it. And then we talked about some, okay, pollen pistil interaction. Always, I have seen multiple times a question coming on pollen pistil interaction. Do you understand what is pollen pistil interaction and why it is important? Just thumbs up if you understand this.
Sir, can you repeat?
Pollen pistil interaction, this thing, this part. Do you understand what is pollen pistil interaction and why it is important? Okay, perfect. All thumbs up. And then double fertilization, triple fusion, these two terms. What is double fertilization? What is triple fusion? Everything is clear here?
No, sir.
No. Okay. So Iram has some conceptual gaps, right? In double. So why do we call some in plants? Why is this name double fertilization is here? Is it clear to you, Parker? So simply remember I told you that there are two male nucleus that moves in a pollen tube. Remember that?
Yes,
And
sir.
somewhere we made a pollen tube. It's not here, but yeah. So there are two male nucleus in a pollen tube, correct?
Yes, sir. One fuses with the eggs and one fuses with the polar nuclei.
Yes, right. So one fuses with the egg cell, which is a single cell, and one fuses with the polar nuclei, which are two nuclei. Basically, there are two nucleus, but both are N and N. Two haploid nucleus is present in the central cell, the biggest cell, and that's how it is seven celled but eight nucleated. And second male gamete fuses with these two nuclei. So together, three haploid nucleus fuse and become 3N, and that give rise to endosperm. And one male nucleus that fused with the egg cell will give rise to embryo. So basically, there are two fertilization events happening. One is egg cell plus male gamete, which give rise to embryo. The other is uh, two polar nuclei plus male gamete, which give rise to a 3N endosperm. That's why it's called a double fertilization event. So is that clear? You know?
Yes, sir.
Yeah. And there is a term called triple fusion. triple fusion is specifically the name for the second fertilization that's happening. The second one, where one male nucleus is fusing with two polar nuclei. If you see, in totality, there are three nuclei fusing. So triple fusion. Make sense? So triple fusion is just a part of the double fertilization event only. Yeah. Any doubts? Aisha,
No, sir.
Iram, Parker. Clear. Give me a thumbs up, Aisha and Parker also. Am I audible? Is there any problem with my voice or something? Okay. No one is okay. Audible. Cool. And then we come to post-fertilization, again, structure and event. So post-fertilization, there are two things that are made structurally. One is endosperm, polar nuclei plus male gamete, 3N endosperm. And then the functions of endosperm, what it does, it provides nutrition. And the second thing is embryo, the male nucleus plus the egg cell. And this is the structure of a embryo, basically. A dicot embryo. This one is a dicot embryo and this is a monocot embryo. We talked about both. The only difference between a dicot and a monocot embryo is that it's all packed in a single cotyledon. Like in case of rice or maize, we have a monocot embryo. In case of gram, pea, and pulses, we have a dicot embryo. So dicotyledon and monocotyledon part was clear, right? Then seed. Yeah, seed. We talked about seed. And yes, this last part also has some questions. False fruits. Any doubt between these terms? Aisha, Iram, Parker. Any conceptual doubt? Yes, no, Iram? No doubts?
No doubt.
Okay. And
So one thing I want to ask, so in this false fruit,
this last part?
the
Yeah.
ye-
Yes, go ahead.
yeah, in the false fruit, the
Mhm.
thalamus and the ovary both will contribute in the fruit formation, or it's just the thalamus. Hello, am I audible?
Yes, it's a good question. Yes.
So your voice.
Yeah, right. Is my voice better now? Am I audible?
Yes, you're audible.
Okay, perfect. I was just changing network. Let me know if my voice breaks. Okay. So to to answer your question, Iram, even if ovary is a part of the edible part ripened, along with ovary, if thalamus is also ripened, okay, or any other part is ripened, then we call it a false fruit. A true fruit is where only the ovary ripens and nothing else. So if something else apart from ovary ripens, it's also a false fruit. And if something along with ovary ripens, that's also a false fruit. Does that make sense? Iram?
Yes.
Okay, that was your doubt.
Yeah.
Yeah. So they call it, they write it here somewhere. Yes. So would you like to read this part? Just read this. What I have made in marked in blue. Iram, just write this part. Just read this part.
Y- Yes, sir. In most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species, such as apple, strawberry, cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruit. Um,
Yes.
most fruits, however, develop only from the ovary and are called true fruit.
True fruits. Yes. So the word also here is crucial. So NCRT speaks in less words, but crisp but clear. So it's not like only thalamus has to ripen so that you will call it false fruit. Even thalamus also, not just thalamus, even if it's peduncle or even if it's some other part, then we call it false fruit. So the true fruit is only from the ovary, if you'll pay attention here. Okay. So these kind of questions might come because they are confusing in entrances also, that a false fruit is option A, where ovary and thalamus both contribute, only ovary contribute, only thalamus contribute, both A and C. Okay. So the answer will be both A and C, because ovary and thalamus both are false fruits. The condition for a true fruit is only ovary.
So,
Are you
what are we
understanding the
actually
concept?
eating?
What are we actually eating
Yeah,
in true fruit?
in false fruit.
In false fruit, it it will be always ovary plus something, or uh, just something without ovary, you know. So most of the false fruit, ovary also ripens a bit. It's not like it does not ripen at all. But majority of the flesh or the pulp comes from, not the flesh, pulp comes from other parts like thalamus. Okay,
Yes, sir.
cool. But one thing is, keep one thing in mind: if there is a fruit where you are only eating ovary, that's a true fruit. Okay. Okay, cool. Any issues with apomixis and polyembryony, these two processes? Iram?
No problem.
And Aisha, what about you? Okay, perfect. So yes. So Iram, going back to that board, do you think so? Anything else from this chapter which is still you find as your weakness, or you are not 100% sure with?
Uh, Actually, sir, I have to revise it once more and then I'll
Oh,
actually
yeah,
come back.
right. Yeah, makes sense. Okay, cool. So do that, and then let me know if there is any other issues with this. Okay. Uh, Wait. Now let's also, since we are doing reproduction, I'll also go to animal reproduction. Yeah. Human reproduction. Can you see the screen that I'm sharing? Aisha, you know?
Yes, sir.
Okay. So in human reproduction, again, we started with system structures basically. So the male reproductive system. So all the anatomical names that are there in this diagram, again, take a good look and try to understand which part is where anatomically, because again, just to label, things can come with missing labels A, B, C, D, and then you have to find out which is the right label. So any doubt with any of the structures here in male reproductive system, take a good time. I'm keeping this figure on board here. just look at the markings of these labels. Don't look at anything in the text part. Just look at these labels. And if you read something like urethra and nothing comes to your mind, you're blank, then we can quickly discuss that thing. Aisha, Hiniram, just take a look. And if everything is, you have understood it, you just think that you need to go back and revise, then you give me a thumbs up. But take a good minute or two. Yes, Iram, it's clear to you. Aisha, what about you? Okay. And yes, then this part of seminiferous tubule. So both of you understand basically what spermatogenesis is. So there were three terms: spermatogenesis, spermiation, and one was
Spermiogenesis.
spermiogenesis. Yes, all these three terms are clear.
Yes.
Iram and Aisha. Yeah, Aisha, to you. Okay, and I will then here only. I would like to ask. Okay, or let's go how the book does that, and then we'll talk. Okay, and then coming to female reproductive part, anything which is conceptually not clear in the female reproductive structures, just look at every label again and tell me if everything is clear. So this is just the anatomical structure, and their functions are mostly, so the labiums are there to just to protection, but pay attention to this diagram. This has a lot of functional labels.
So,
Any doubt? No doubts. Okay.
so,
Yes, Iram.
so do they ask us to draw these diagrams?
They asked, like, I remember asking this kind of, it came in when I gave the boards, of course. They asked to draw and then label. I'm sure that in boards they can. Okay, in boards they can literally ask you to draw from scratch and then label it. Okay, but mostly in other exams in entrances, you will get the diagram and it will all be about one label which is mislabeled or something like that. But yes, in boards they do ask to make diagrams. But then they will ask diagrams which are more conceptually driven. Like I've never seen them asking the sectional view of the female reproductive system, but always the diagram, this view will be asked, where you can label everything like the ovary and the fundus and all. So don't worry about this diagram at all. Similarly, in the case of male, this diagram is not asked much, which is the whole anatomical part, but only this view of the male reproductive system. Okay, you know,
Yes, sir.
just don't worry too much about it. If it, just take a good look and remember which part is where. And even if you miss one or two labels, like you managed to label fallopian tube, but you didn't label isthmus, ampulla, and infundibulum, uh, and specifically where they are, it's okay. And the main thing is, if you missed uterine fundus, it's fine, but you shouldn't miss the cervix or the ovary or the endometrium, because these are important markings. So just like that, if you manage to mark everything, it's good. Okay. Same as with the mammary gland. Again, remember then that the part ampulla is both in the mammary gland and also in the fallopian tube. So if something comes about ampulla in the question, read the question carefully. Which ampulla they are asking about, of the female reproductive system or the mammary gland. Okay. Okay, now coming to some mechanisms. So gametogenesis, first spermatogenesis. Any doubts in spermatogenesis?
So I have a doubt in this part, the
Yes, go
mammary
ahead.
gland. So
Mammary
it
gland.
says
Okay.
that
Mhm.
during pregnancy the cells will differentiate, and
Mhm.
after the parturition they'll be able to produce milk, right?
Right.
This is what it says. So how long does that uh, last? Like, is it 1 to 2 years till the breastfeeding, or is it like it does stop or not?
Yes. So good question. So there are two things associated. One is differentiation. So remember, differentiation is where the mammary glands in females achieve maturation, right? Structural maturation. So when they talk about differentiation, they are mostly talking about these alveoli. So you see a mammary lobe. There are 2, 3 lobes here. In each lobe, there are lots of mammary alveolus. Just like there are alveoli in the lungs, there are these small um, cavities. Here in this case, they are fluid filled. In case of lungs, they are air filled. So fluid is the colostrum. So they basically synthesize and secrete the colostrum, the milk. So they differentiate during the pregnancy. Once they have differentiated and mammary glands are all structurally ready and mature, the differentiation does not go back. It cannot, undifferentiate again. It will always stay differentiated, but the ability of the female to synthesize colostrum, that stops after, a year after parturition. Year after parturition. So after parturition, the female starts secreting, synthesizing and secreting milk. But over time, that synthesizing and secretion of milk ability will start declining and declining. And that is where mothers have to get their kids off breastfeeding. But still, when if the female conceives next time, she's again pregnant, then again, the mammary glands are differentiated. It will again differentiate a little bit more, but mostly it's already differentiated in the first pregnancy, and it's ready to synthesize and secrete colostrum again. Does that make sense, Iram? So
Yes.
differentiation word is for proliferation of these mammary alveolus. Okay. And functionality part is where it synthesizes colostrum and secretes it.
Yeah.
Yeah. Okay. Cool. Okay, perfect. Any doubts in gametogenesis? All these cells, Sertoli cells, spermatogonium cells, primary spermatocyte, secondary spermatocyte, their ploidy levels, everything is clear, Aisha and Iram?
Yes.
Okay, very well. Going forward. Yes, about the sperm. About the sperm, just remember some major things. One is the neck piece is very important to propel, like to provide energy for propelling the sperm in a whiplash motion. The tail is mostly the part which produces that motion, and head is the major part that contributes during fertilization. and head is the only part that enters inside the ovum, and all the neck and the tail stays out. Okay. And then what is the energy source for sperm? That is not glucose, that's fructose. That's why semen is rich in fructose. And seminal vesicles are the glands that produce and release fructose into the semen. Okay. These are some major points you should remember. Now coming to oogenesis. Any doubts and questions in oogenesis process? Think about it and then tell me. And this schematic representation where they have parallelly compared both. What is corpus luteum? What does it secrete? Graphian follicle, antrum, all these terms. Okay, it's clear to Iram. What about you, Aisha? Okay, clear to you as well. Perfect. So coming to menstrual cycle, this graph that summarizes it all. So remember, when we talk about menstrual cycle, there are two organs involved, the ovary and the uterus. So there are ovarian events and there are uterine events, and then there are two sets of hormone also. One's first set of hormone, which is secreted from brain, pituitary, they act on ovaries, and then the hormones that are secreted from ovaries, they act on uterus. Okay. So all these four things along with their progression, just take a look at this diagram and let me know. We also made it during the class from the scratch, but this is more properly made. Let me know if there is any doubt. This diagram, they can ask you to label in entrances. And I've seen many questions coming from this diagram. So you have to understand this one really, really well because it's a mechanistic diagram. It's not structural. Give me a thumbs up if all is set. Okay. Aisha? Okay, perfect. And then uh, it goes for fertilization and implantation. Remember these layers. So they ask you from outer to inner, what are the layers that surrounds the ovum? So corona radiata is at the outermost, then zona pellucida, and then the pre-vitelline space, and then the membrane of the ovum, plasma membrane of the ovum. So corona radiata, zona pellucida, the space between zona pellucida and the membrane, which is called pre-vitelline space, and then the membrane of the oocyte. Okay, and then inside is the cytoplasm. Just remember this. Yes, the concept of polar bodies, is it clear? I'll come back to this part here. Sorry, we missed it. So remember, there is this first polar body and second polar body formed. So that's the difference in case of male and female gametogenesis. Is that clear, Aisha and Iram? Okay, perfect. And then menstrual cycle done. This is done. Yeah, in case of implantation, just remember these names: morula, blastula, blastocyst. Okay, so first it's a single-celled zygote. We don't call it embryo. It becomes, when it starts dividing, the first division makes it a two-celled stage, which from here onwards we call it an embryo. And morula is anything um, around 8 to 16 cell stage is called morula. And then each cell becomes blastomere. Okay, each daughter cell is called blastomere. In a morula, there are 8 to 16. And then blastula is where it comes out. So morula stays in the fallopian tube. At the stage of blastula, it enters into the uterine space. And as a blastocyst, it is implanted. So remember that blastocyst is the stage where implantation happens, not at morula or blastula. And blastocyst has two layers: the trophoblast, which is the outer layer, which you see here in purple, and the inner cell mass. Also remember that trophoblast does not give rise to fetus. It's the inner cell mass that gives rise to fetus. Trophoblast makes the what you call um, placenta. Okay, Iram and Aisha? Perfect. Then pregnancy and embryonic development. This is all at which month. For first, what hormones are important. So here you have to remember fetal ejection reflex. Before that, when the pregnancy is ongoing and the embryo is developing, placenta is the first structural and functional connection between mother and the developing embryo, how it is formed. And once it is formed from chorionic villi from on the trophoblast, the outer layer that we talked about. And once it is formed, then it starts secreting hormones like these ones, which are here in the marked here. Okay. HCG, human chorionic gonadotrophin. And what is special about HCG? Anyone remember? Aisha, do you remember, or Iram? What is special about HCG? What is HCG? It is one hormone, the presence of which in the body fluid of a female confirms pregnancy. So it's also called pregnancy hormone. All the pregnancy kits where one drop of female urine is tested, it tests for the presence of HCG in the urine. If there is HCG, it's for sure pregnancy positive. So HCG is pregnancy hormone. Okay. Is it clear?
Yeah.
Yeah. And then relaxin, though it is written here, but it is not secreted by placenta, it's secreted by ovary majorly. And it helps in, as its name suggests, relaxin. It helps in relaxing the muscles of the uterus, the endometrium, and helping it as the uterus becomes bigger and bigger and bigger, helps the female with proper extension of it, so there is no pain. Okay. And just read these parts. And yeah, parturition and lactation is all about, parturition is childbirth. And sorry, in embryonic development, you also have to remember during which month or trimester what happens, right? So I dictated these to you, that in first month what comes up, in the second month what happens. And just remember that by the time the second trimester ends, the fetus is structurally complete. It's done. in the last trimester, which is month 7th, 8th, and 9th, nothing new is formed, but everything gets better, or it's proper. Everything completes. Okay, but if you'll see, nothing new structurally is formed in the fetus in the third trimester. The third trimester is all about making the fetus mature. That's why even when there are premature births in the 7th month, sometimes the fetus survives, the baby survives and can grow up to become completely normal. Okay.
So what's the reason behind it?
Yeah. So what's the reason behind what? Behind the
Immature
premature?
babies.
Yeah, right.
Premature.
So premature birth sometimes happens due to there are two, or there's not a single reason. There are multiple reasons. One is if the female's body is not capable of continuing with the pregnancy, or it starts taking a toll, then the female body would like to expel out the fetus. So that fetal ejection reflex is just induced without the placenta or fetus telling to do so. That is one case. That's called accidental fetal ejection reflex. Or sometimes due to hormonal imbalance also during pregnancy, especially when the mother is malnourished, the fetal ejection reflex happens earlier than it should happen. So that these are the two probabilities. Yeah.
Yeah.
Basically, when oxytocin starts coming in from the brain, from the posterior pituitary, remember anterior pituitary and posterior pituitary? From posterior pituitary, there were two hormones, oxytocin and vasopressin. So oxytocin should only come when fetal ejection reflex is given by the placenta and fetus to the brain. But if due to hormonal imbalance, if it happens by the end of 6th month or 7th month or 8th month, anything before 9 months is called premature delivery. Then whenever oxytocin will come up, it will start, vigorous contractions of uterus, and the childbirth has to happen then. Yeah. And for the 9 months, what keeps the baby sealed? Because there is fluid all over the baby. We call it amniotic fluid. And it's like a sealed bag till for 9 months. That is sealed because of a plug of mucus in the cervix. So it stays tightly packed. And when that plug breaks, that's what is called as the water breaking event. That for a pregnant female in the 9th month or towards the end, you must have heard the term the water broke, and the female, that is first sign that the female is ready to give birth in few hours now. Yeah. Okay. Anything, any doubt in parturition and lactation, the last part? No.
No, sir.
Okay. um mm, okay, then it's over. So in human reproduction, there is no one has a doubt. I showed you. Before I go for some questions, let's also yes, reproductive health. This is the last. So we'll finish this one unit. I just want to make sure that you all are on the same page. Yep. Can you all see the reproductive health chapter?
Yes, sir.
Please utilize this class if you have missed anything. I assure you. Yeah. So reproductive health starts with First thing is population, family planning. All these are theoretical things we talked about. Some facts to remember that family planning started in 1951 with the Family Planning Commission being set up in India. And the whole idea was to have a reproductive and child health care program for better health of the mother, the baby, and also for population control, spreading awareness for population control and uh, gender related issues, sex related aspects. Okay, hygienic sexual practices. So this is basically what was the aim. If you read here, uh, why is so sometimes this kind of question comes in board, then why is sex education important to be introduced in schools, etc. um, so yes, so Iram, will you read this part that I have highlighted quickly so that we can then move over? Iram, are you there, Iram?
Yes, sir.
Yeah, thank you.
Introduction of sex education in schools should also be encouraged to provide right information to the young so as to discourage children from believing in myths and having misconception about sex related aspects. Proper information about reproductive organs, adolescents, and related changes, safe and hygienic sexual practices, sexually transmitted diseases, STD, AIDS, etc., would help people, especially those in the adolescence age group, lead a reproductively healthy life.
Mhm.
Educating people, especially fertile couples and those in marriageable age groups, about available birth control options, care for pregnant mothers, postnatal care of the mother and child, importance of breastfeeding, equal opportunities for the male and the female child, etc., would address the importance of bringing up socially conscious, healthy families of desired size. Awareness of problems due to uncontrolled population growth, social evils like sex abuse and sex related crime, etc., need to be created to enable people to think and take up necessary steps to prevent them and thereby build up a socially responsible and healthy society.
Yeah. So everything, like, it's a very broad umbrella. And why sex education is important, you can just s- break this whole paragraph into points. And if this question comes, take it as a bounty question and just get good marks. Okay. These kind of questions are always included because the way board paper is set, they have to decide a minimum difficulty criteria, minimum difficulty level, so or maximum difficulty level, how much. So that is basically to ensure that even if someone has worked decently uh, enough to study the subject, they should all pass. So one thing that boards often focus on is passing students leniently, but then to be among the cream or to be among the toppers, you have to work really hard. So it's easy to pass, but slightly it requires slightly more meticulous efforts to go beyond 85, 90%. So that's it. So these kind of questions will be there, keeping in mind students who just want to get passing marks, and you all can take advantage of it because I'm sure you are all going to do great. Yes. How was your pre-board, Iram? Your pre-board has happened.
Yes,
How
pre-board
was it?
1.
Oh,
It was
there are multiple pre-boards
this
now.
pre-board 1 and 2. The
One and
second
two.
one
Okay.
is after I think after
So
January 7th.
so how did they? So both the pre-boards have cumulative syllabus, the complete syllabus, or they divided it into two?
Complete syllabus.
Okay, that's kind of good, no? You get two
Yeah,
practice tests
and
before. Yeah, how was the first
yes,
one?
it was okay. Like I scored 59 out of 70, which was not expected. Like I really worked hard, but actually the thing is that in the first term one examination, I got 68 out of 70, and then it dropped to 59. Wasn't expecting, but yeah, I'll
But
work
then
hard.
in term one, was it full syllabus?
No, it was just 6
Right.
chapters.
So
Yeah.
right. So I don't think it's too bad. See, it's just looking bad because of number. 59. When the total has a 7, looks like, oh, you didn't do good. But imagine if it was 61, you would not have taken it so bad, right? 61 out of 70 is like, oh, just 9 marks.
Yeah.
But 2 more marks make psychologically more difference. Yeah. It's like, yeah, it's like you have your, yeah, that happens in sports as well. Also, like in cricket, they say that if you're chasing down anything below 200 is psychological, you have a psychological advantage. But anything above 200 is like, oh, 200 plus. So 11 marks short, but which is like you can easily grab 7, 8 more marks, right? To me, you have potential. So don't worry about it. When is your second reboot? January.
Yeah, it's estimated to be around January. Yeah.
Okay, cool. So throughout January, we will be mm, focusing only on and solving problems. So hopefully that will also help. Okay. Um, Everything clear with amniocentesis, the concept of amniocentesis, how it works, and why it is banned in developing countries like India and many other places. In fact, any doubt here? Amniocentesis and how it works? Aisha,
No.
Aisha, how was your pre-board though? Aisha, you were not able to speak, so but you can write if you want to share. Are you satisfied with your performance in pre-board, or do you think something needs to be changed? Didn't work. Okay, and Aisha is saying it was fine, but not the best. So I think Aisha and Iram just want to say the same thing. Genetics part was a little weak. Okay, so after this we'll come to genetics. So no worries. Okay, don't worry. Uh, did you miss any classes, regular classes of genetics? My lectures. If you
So,
did, why am yes, Iram, go ahead.
so I actually did, because the time you were teaching genetics, the
Mm-hmm.
first chapter, I was on vacation, so I missed like
Oh,
around four or
okay,
five classes. Yeah.
okay. Yeah. So even Aisha is saying that she was in India that time, so I think she missed few parts. I think both of you were going through the same phase. Yeah, cool. So do two things. First is the whole repertoire of all my lectures is on Learnivio's YouTube channel. So all 72, I think now it will be 73 or 74 videos are there. You just have to look for 12th biology and my thing will pop up and just go there. It has some 72, 73 videos. Look for the topic that you think is weak or you missed and go through it at any given time of your day, okay, as a revision. And then you can come up with your questions. And since because our classes are limited to days and time, we have a WhatsApp group which now can be utilized the best. I always reply. You send me a message anytime. You both are on the group, right? Let me just see if you are there. Yes, right. I can see Iram there. I'm not sure about. I can see Aisha also there. So at any given point of time while listening the lecture, if you have a doubt or if you have a question, just type that question and send it on the WhatsApp group. Yeah, the group's name is we had Rahul Sir G 2023-24. You both are on that group, right? Yes. So please um, make use of that group. Till now I'm using that group to publish announcements and notices and things like that. So now it's your time. Use it, send questions, and that way if Parker has a question, or if Aisha has a question, or if Iram has a question, everyone can see it and I'll reply with answers or potential things what you need to read, and then everyone can benefit at the same time. Okay, so collective efforts and collective benefit. So let's make that group a little more vocal and active now. Okay, so two things, and then of course we will discuss in class, no problem. Okay, so the birth control methods is a very important part of this chapter, reproductive health from entrance perspective. Okay, even in boards they ask, but the question will be like, talk about or write about briefly natural methods or barrier methods. So you can write, but the details become important for entrance questions. So you have to be very meticulous in understanding the names of the natural methods, the name of the barrier methods, which methods can prevent STDs and which cannot. And for that matter, only one method can prevent sexually transmitted diseases from happening and no other method. And that one method is a barrier method, which is using condoms or femidums. So femidum is not that popular, but it's basically the same thing. It's a female condom. It's inverted in its structure, because it is a latex sheath that does not allow the body fluids to mix. The moment there is body fluid mixing, even if there is no fertilization, there the sexually transmitted diseases will spread. Okay, so other methods only prevent fertilization, but the barrier method, condom and femidums, prevent fertilization plus STD both. So just remember some of these facts. Name of these instruments, like IUDs. So what kind of IUDs is what? So especially this part here.
So,
Yes, Hiram, go ahead.
so when we talk about sexually transmitted disease, is it like
Mhm.
only a female is confined to have a disease, or is it like both male and female are
No,
likely to have it?
both. No, both. So all sexually transmitted diseases can be transmitted from a male to a female and can also be transmitted from a female to a male, because during the act of sexual reproduction, the coitus, the body fluids are mixing. The mixing is happening for sure in the female's reproductive tract, but that does not mean that male cannot get that disease. Males can also get that disease. Okay, it's not only confined to female system. Make sense, you know?
Yes, yes.
Yeah, yeah. Only fertilization and pregnancy, for obvious reasons, it can only happen to female. Okay, and not to males. But viruses, bacteria, pathogens that cause disease will affect the organ, the penis, and also the uterus or the vaginal tract equally. Okay, so just pay attention to this paragraph, which I have put here in blue. It talks about exact examples of IUDs that are non-medicated and medicated. Medicated means they release some hormones, and non-medicated means no release or no hormone is involved. There is only one non-medicated, which is lips loop. And apart from lips loop, all other are medicated, either copper releasing or hormone releasing. Okay. And so number both. Okay, cool. Then oral method. It's important because India made an advancement in this. They made a pill called Saheli, which is once a week pill, and it is non-steroidal preparation. Okay, and that's where the contraceptive value is high and the side effects are low. So that's one important thing to keep in mind. Yeah, and oral administration or oral contraceptive is very popular in among females, but the most effective or the most long-term uh, uh, functional is IUDs. Okay. Okay, cool. Fine. Yes. And when we talk about progesterone, so all these contraceptives work on the principle of your um, the principle that you studied in menstrual cycle. So remember, what does progesterone do? The hormone progesterone, Aisha, you know,
It inhibits ovulation.
inhibits what? Uh, in fact, yes, you're right, but it does not work on the ovaries, right? Ovulation happens from ovaries. You remember that? And the effect of progesterone is on the uterus mainly, if you see. So it maintains the endometrium. Okay. And if it maintains the endometrium, it gives a signal to the brain. So when is endometrium maintained? Normally, in a normal reproductive cycle of a female, when will endometrium be maintained? When pregnancy will occur, right?
Yes.
When fertilization has happened and implantation is about to happen, you will not like the endometrium to break down now. So it will be maintained. So giving progesterone from outside tricks the female reproductive system and the brain that, oh, you don't have to do another ovulation because we have to maintain the endometrium. So that's how they work. So if you use progesterone and put it under the skin as implants, where for very long time they keep secreting it slowly and slowly, they will be very effective for very longer periods uh, to avoid pregnancy. And of course, they also affect ovulation, like you said, in the process. That's right. MTP, again, it's simple medical termination of pregnancy, or also known as abortion. We don't call it abortion, but induced abortion. So the term induced abortion of implies that it was induced for a reason, and it can only be done in the first trimester. And once the female has reached second trimester, it shouldn't be done unless and until there is some rare case for which you need medical. These sexually transmitted infections, um, there are a bunch: hepatitis, HIV, all these, and how to avoid it. These are all general knowledge things now for you. And yes, then this part, infertility part, from here to here. It's very important paragraph. All these names, all these different methods of assisted reproductive technologies is very important. Okay, Aisha, Iram. So if I ask you, okay, let's do one thing. Let me pull out some important questions. So read, I'm giving you 3 minutes, but to both of you, read whatever I have highlighted here in blue, and then I'll ask questions, some questions, testing the concepts. Okay, Iram, Aisha, give me a thumbs up if you are reading. Perfect. I'll give you 3 minutes of silence.
Done, sir.
Okay, what about Aisha? Done, Aisha? Perfect. So now the kind of questions that are asked are like this. So you understood IVF and all these things. So major thing is, when do you think that ZIFT is preferred over the gift part? The zygote intrafallopian transfer is more preferable over the gamete intrafallopian transfer. In what condition? So these kind of questions where, so if the zygote intrafallopian transfer means to make the zygote, the fertilization, where that happened? Outside, right? That's how you are implanting, or you're transferring the zygote back into the fallopian tube. So if I am transferring, or if any doctor is transferring zygote back into the female system, the zygote has to go into the fallopian tube. Okay. And if it is embryo, completely like more than one cell stage or few cell stage
Uterus.
embryo, where that, yes, that should go into the uterus, right? So that ZIFT is preferred over the gamete intrafallopian transfer, where the female is also not capable of fertilize, like conceiving the sperm, or the sperms are not able to survive in females. So female's body either is too hostile, or the female's fallopian tube is not very receptive to sperm, so that fertilization can happen there. And what is the natural site of fertilization in a female system? What is the site of
Ampulla
fertilization?
isthmus junction.
Perfect. So ampulla isthmus junction is quite some long journey from the uterus, right? They have to go, and so first thing is females are more hostile, and the second thing is where the male partner is not producing enough sperms. So per ejaculation, there has to be somewhere around 30 to 300 million sperms. Okay, that's a huge range. But if it is less than that, then the chances of sperms reaching ampulla isthmus junction and to be able to find a ovum and fertilize it becomes very low. So trials after trials, the couple fail to conceive. In that case, ZIFT is the most preferred way, where you take out sperm from the male system, take out eggs from the female system, do a IVF, and then transfer the zygote. Okay. But when is gift preferred? Gamete intrafallopian transfer?
When the female is not able to produce. So.
Yes. So in that case, the male is fine. The male is also producing enough number of sperms. The female is also fine. Like the anatomy is completely fine. She's not very hostile, or the problem is not because sperms are not surviving. The problem is because either the female is not able to secrete ovum properly, or the fimbriae or the fallopian tube is not working properly to trap the ovum when it is released from the ovary, or something is wrong with the ovaries and ovaries are not producing proper oocytes or releasing it at the time, right? In all these cases, we can take another female's ovum, and then just at the time where ovulation should happen normally, inject it into the recipient female's fallopian tube, and then the couple can normally go and copulate, and fertilization will happen, and implantation will also happen normally. Yeah. And then when is ICSI, intracytoplasmic sperm injection, is preferred.
When the male is not able to produce milk.
Yes, when the female system is completely fine, even the male, no, not only when the male is not able to produce sperm. If the male is not able to produce sperm, where are you taking the sperms from, right? So even if the male is able to produce sperm, there are disorders like erectile dysfunction in males, where the tissues of the penile structure, the corpus spongiosum and the um, cavernosa, the tissues do not undergo erectile uh, form, right? And it's very important so that the sperms are transferred or transported right up to the door of the vaginal canal where the cervix starts. So if that's not happening, and if most of the sperms are just, dying, then in that case, sorry. No, we are confusing it with, oh no. Yes, you're right. It's, I'm confusing it with artificial insemination, sorry, or intrauterine insemination. Intracytoplasmic is when you take a single sperm nucleus and just inject it into the cytoplasm of the ovum, right?
Yeah, fine.
Yeah, it's just like IVF, but one step ahead of IVF, where in IVF we put we put a ovum and we allow a sperm to fertilize it. So thus still the fertilization is being done by the sperm, but yes, we are facilitating that. But ICSI is where you're right. Either the male is not producing um, female, not producing enough sperms to fertilize the female egg, or in cases where the sperms are not getting properly matured, they are not attaining functional maturity. So there are lots of sperm, but none of them are capable of swimming with that much of energy so that they can naturally go and fertilize. So in that case, all you have to do is not even take the sperm, but sometimes just take the head or the nucleus also and inject it into the female uh, oocyte. So intracytoplasmic. And what I mistakenly got confused with was intrauterine insemination or artificial insemination. These two are where the sperm count is very low or the sperms are not very healthy. So if they are not very healthy and also their number is low, then you would like to drop them right at the door of the uterus. So intrauterine, it has to go deep inside the uterus where you leave them so that all they have to travel is that arm of the fallopian tube. But if they can, move okay, their health is okay, but they are low in number, then it is collected from the male over multiple donations, multiple times, and then inseminated artificially in the vagina. Okay, cervix. So for selecting which kind of assisted reproductive technique that the couple needs, first the male and the female both have to go through examinations. The male will be examined for his sperm quality and quantity and the health, and the female will be examined for her ability to produce, release, and allow the movement of sperms. So body fluid from the vagina or from the cervix or uterus is also taken to examine whether it is too hostile, too acidic, and also if they are synthesizing and secreting at the right time or not. Okay, and also in cases where females fallopian tube is not able to facilitate the movement of ovum down. So if it enters the fallopian tube but cannot come down to ampulla isthmus junction at the right time, again it's the issue with the female tract. So in that case, you can do ZIFT. Okay, make sense, Aisha, So the kind of questions that can come from this part is they will give you a situation and then they will ask you which is the most optimal solution or most optimal a- assisted reproductive technique that will be used in this condition. So you have to read the question carefully and decide for yourself what basically is lagging and which of this can serve or solve the problem. Is it okay? Aisha and make sense? Okay. So just read this again, and just like you did now, when you are reading it in your own notebooks, you can keep shorthand notes. Then this will be conditions, technique. So under what
So,
condition, what technique you will select? Yes, Hiran, go ahead.
so when you said this case where the female body is very hostile of, producing
Mhm.
ovums.
Yeah.
So, is she even able to, hold pregnancy up to 9 months?
Yeah. So if the whole uterus is hostile to implantation, then none of this ART will work. Then the female has to go for surrogacy. Surrogacy is another, like, it's not assisted reproductive tech. I think it does come under assisted reproductive technology, but then it's like uh, using another woman's womb. But in that case, the ovum and sperm will be of the biological parents. So that condition also happens actually. But since surrogacy is not in your syllabus, right? It's nowhere discussed in
No, it's not
NCRT,
that.
no? Yeah, so I don't think they will give you that in the options. But what you are asking also can happen.
Okay.
Okay. Yeah, cool. Good question. Yeah, okay. So this brings us to a final round of revision for reproduction unit along with reproductive health. And you have 1 week next Wednesday. So I think Monday will not have the class because of Christmas falling on a Monday, right? 25th is Monday. Am I right?
Yes, sir.
Yeah. So Wednesday we'll meet and we'll do the same thing with questions for genetics. Genetics means chapter 5, 6, and 7. I will also take evolution, just like we did 3 chapters here. And then both these major mammoths chapters units will be done, and then we can smoothly move towards just problem solving mode in January, whatever classes we get. Okay, Aisha and Iram,
Yes.
does that
So
sound
it's
good?
better for us to revise the chapters because it's useful for revision. And then we can say, what the doubt is about in the chapter.
So yeah, right. So you're suggesting that it's better that next Wednesday we revise
No,
genetics,
no, no.
right?
We
No,
revise. No, no,
you
not
revise
in the
genetics.
class. Yeah.
Okay. Oh, yes, right. If you do that, then it will be great. If you revise it and then come to the class, it will be great. We can focus more on questions.
Yes, sir. That's what I mean.